Well, good morning, AP Bio students. Mrs. Shelton here. Just going to talk to you about one of my favorite topics, Chapter 13, Molecular Basis of Inheritance. What we're going to be learning about today is actually what you already know. You already know the basic structure of DNA, but we need to understand actually the scientists who really contributed to our understanding of DNA structure. But most of what today is going to be about is talking about all the enzymes that are involved in replication of DNA, including these enzymes here in the you must knows. And then also understanding then a little bit more about chromatin through chromosomes in our DNA packaging and how that actually affects then gene expression when DNA is packed really tightly or when it's packed very loosely. Um, what I want to point out to you on your notes, though, is there's actually two sections of You Must Knows from the test prep review book. In 13.4, it's understanding our genetic engineering or biotechnology. So here's some You Must Knows from this. Now, you don't need to know the details of this. This is why I also clip arted for you just what College Board is expecting you guys to know here in biotech. Just be familiar with what the purpose of some of these different types of genetic engineering or biotech tools actually are, and really relating it to our understanding of DNA replication and DNA structure is what you would be doing. So let's actually flip over here to our PowerPoint. Let me see if I can get this presentation to work for us so we can see it on the big screen where I can change it so we can view it full. Hide presenter view, and let's see if that works. Ta-da! We got it to work this time. Okay, so, oh, no, we didn't. Let's go back here. Okay, so DNA, here you go. Here is your double helix. So you've already seen this structure lots and lots of times. What we now need to make sure that we are familiar with is understanding this double helix. What is making up this backbone? What is making up then these rungs of the ladder in this double helix, making sure we're understanding our base pairing rules, the hydrogen bond interaction that's here. Also then how DNA actually replicates itself is what we need to do. Now, it actually wasn't until like the 1950s that we as scientists, not we, because I wasn't alive then, but this is actually new knowledge in the scientific community as far as DNA and understanding that DNA is our genetic material. We kind of take it for granted that this is something we've always known. And then when you actually look at how far we've come with DNA technology in just a really short period of time, it's pretty remarkable as far as what we have been able to discover and determine and lead to in our understanding of just what controls our gene expression. So let's kind of get to a little bit of the history of understanding how we determined that DNA was our genetic material. And one of the scientists that you first need to make sure that you are aware of is Griffith. Now, Griffith, he did an experiment. I call it Griffith's dead mouse experiment. That is not what the official name is, but from this picture, it always reminds you of, okay, Griffith did the dead mouse. He actually did, and what he determined is that there's a molecule that can be inherited that can transform a cell. So he actually did the transformation study is what this is actually referred to. Now, transformation means that the properties of that cell have changed, but it's a molecule, and the incorporation of a new type of molecule actually allows for this transformation. So in this study, what he did is he actually took a type of cell, this living S cell, and when injecting that into the mouse, it was lethal to the mouse. So this was an infectious agent that killed the mouse. Now he took another type of cell, an R cell, and when he injected that into the mouse, that was not lethal to the mouse. So I've got a deadly cell, I've got a non-lethal deadly cell injected into the mouse. Now, then what he did is he took this deadly cell, this S cell, and he heat killed it. So it was no longer a living pathogen, but the molecules were intact. And so with that, when he injected just this cell alone into the mouse survived. But then, so this is really what the experiment was all about, is he took this heat-killed S cell with all of its molecular components, but not alive, and he mixed it with this non-lethal cell. And when he mixed them together and injected it in the mouse died. Well, then what he did is he took out the cells to find out what was the deadly pathogen. And actually, what he found is that these cells had transformed. They had changed their properties to now be lethal, is actually what had happened because of a transfer of information. Some type of molecular information from these to these led to this transformation. So then began this race in the scientific community like, okay, molecules are the transforming agents. Well, then it was, what is the transforming agent? Well, we knew macromolecules were very complicated. Remember, with our four macromolecules: carbs, lipids, proteins, nucleic acids. Well, carbohydrates just can't explain the complex information that our genetic information has to have. They're very, and even though they can be long, there's not a lot of variation within carbohydrates to be our genetic information that contributes to all these differences in living things. So that was known, that carbohydrates were not the transforming molecule. Now, it was also known then that lipids just don't have that diversity either to be able to explain the genetic material that gives the diversity of life as well. So really the argument that was being had within the scientific community was, is it proteins that's our genetic material that we pass on, or is it DNA? Now, actually, in the scientific community, they didn't think DNA was the transforming agent because it was so simple. There's only four nucleotides: G, C, A, and T. But with proteins, actually kind of a valid argument was, wait, there's 20 different amino acids that can all combine in lots of different forms, so maybe it's proteins. Well, Hershey and Chase did an experiment that actually proved that DNA is the transforming molecule. It's DNA that gets passed that can now transform the cell and give it different characteristics. So what they did in order to study and to prove this is they actually used bacteriophages. Now, a bacteriophage, this looks like a little alien. A bacteriophage is a virus that infects bacteria. So this is a virus. Now, a virus is not a living molecule, but it is genetic material. And in this case, bacteriophages have DNA, and then they have a protein phospholipid outer capsid is what they end up having. So since it's really protein and DNA, it's the perfect thing to use, because they knew that a bacteriophage could actually transform then the bacterial cell that it infects. So what they tried to do is to determine, is it the proteins that stay and get injected into the bacterial cell, or is it the DNA that is the transforming agent? So what they did is they radioactively tagged bacteriophages first with sulfur. Now, the sulfur, what we have to remember is sulfur is found in proteins. So the sulfur, remember that sulfhydryl group that is one of the R groups in an amino acid. So the radioactive sulfur allowed them to track then where the protein was going from the bacteriophage into the cell it was infecting. Then they had another batch where they now radioactively tagged the bacteriophage with phosphorus. Now remember, phosphorus is a part of DNA, but not a part of proteins. So this radioactively tagged phosphorus is actually what was found then inside the transformed cell. And so it was because that it was the radioactive phosphorus that proved that DNA was the transforming agent. Now, once they kind of proved that, there was other scientists that were also proving the exact same thing. I mean, so this is not just, one person did it and, well, everyone believes it. I mean, scientific community likes backup. So there's definitely plenty of other scientists doing the same thing. So we had Hershey and Chase, but we also had Avery, McLeod, and McCarty, who also proved that DNA was the transforming agent as well. But really, this would be the one to make sure that you're understanding the Hershey-Chase experiment with a radioactively tagged protein or radioactively tagged DNA is the one to make sure that you're understanding. Now, within the scientific community was like, okay, DNA with only 4 nucleotides, because this was the basic nucleotide. They knew the basic nucleotide structure was a pentose sugar, a phosphate group, and a nitrogenous base. So they knew that this was the nucleotide, and there were only 4 nucleotides in DNA. So the race began to kind of figure out what's the structure that now can explain this huge diversity we see from one living thing to the next in this DNA when there's only 4 nucleotides. So a couple of scientists to be aware of in order to learn the structure of DNA. We should know that there was a scientist named Chargaff. Now, Shargoff actually determined through analysis that there was always an equal amount. So in any DNA sample that he ever took, there was always an equal amount of G's to C's. So if I had 30% of my DNA was guanine, well, that meant that I also had 30% then that was cytosine. Well, if you actually think about that, if G and C always are equal in their quantity, and then A and T are also always equal in their quantity, if I know the quantity of one, I actually know the quantity of all four of my nitrogenous bases. So that Shargoff's rule is that the quantity of guanines and cytosines are always equal. The quantity then of thymines and adenines are always equal, regardless of the DNA sample. Now, then came along these handsome devils here. So this is Watson and Crick. So Watson and Crick actually are accredited with discovering the official structure of DNA, the double helix. Now, they did not work alone. They had a lot of background information. They knew nucleotides, so they already knew the structure. They knew Shargoff's principle that G and C and A and T had to match. But they're trying to figure out, like, more in just a single polymer is what they were trying to figure out. But then actually they happened to be working with this woman who was at Cambridge at the same time, Rosalind Franklin, who was actually studying structures of complex molecules through X-ray diffraction. And so she actually happened to have this X-ray crystallography picture of DNA. And through this X-ray crystallography picture of DNA, she was actually able to lead Watson and Crick to the idea that this is actually a double helix. So it's actually a double-stranded molecule that spirals around. So unfortunately, because Rosalind Franklin was actually studying molecules using X-ray, we didn't understand the mutagen effect of X-rays at the time. She ended up dying due to cancer and X-ray poisoning. But she ended up being named as part of the contributors in the Nobel Prize winning effort of determining the structure of DNA. We often accredit it to just Watson and Crick, but Rosalind Franklin, actually without her contribution, who knows who would have actually figured out the structure of DNA. Now, what we know about DNA is that DNA is a double helix. That means it is actually two polynucleotides. But they're anti-parallel. This was actually the big aha moment for Watson and Crick is, okay, we know it's two sides and it's in a helix, but how does it fit together? Because they did know, they had all the puzzle pieces. They knew that the backbone side of it was the phosphate sugar. These would covalently bond. And then the nitrogenous bases, though, in order for them to actually then hydrogen bond with other nitrogenous bases, you had to flip one side upside down. So with this is where we have our directionality, and it's absolutely key in this chapter that you remember directionality of DNA. And so for directionality, I need you guys to always remember my 5' end, and because this phosphate group is on my 5th carbon of my pentose sugar, that's why it's referred to as the 5' end. Now, if you don't remember that, just remember phosphate 5, F and F. So wherever I have a phosphate group extending up from my nucleotide, the last nucleotide, that's my 5' end. And on the other side, though, I'm going to have a hydroxyl group on my 3rd carbon of my pentose sugar. This is my 3' end. Now, this is really important when we start talking about building DNA or building RNA, because in building more DNA, we can only attach new nucleotides to a 3' end. That's the only way the enzymes can actually elongate any DNA molecule. Now, the other side then, it's anti-parallel. And being anti-parallel, my 5' end is on the opposite side. But what happens in the middle is my nitrogenous bases, A and T, and G and C meet in the middle. And what we find is with complementary base pairing rules is T and A, they make 2 hydrogen bonds between them. So this is what makes them complementary. G and C make 3 hydrogen bonds between them, which is what makes them complementary as well. So these are my base pairing rules. Remember, we learned GCAT, and actually we learned GCAT puppy puppy 32 as our mnemonic. G and C, A and T always go together. And also remembering that when we take a look at these nitrogenous bases, I have a double ring structure, that's a purine. always bonded then with the pyrimidine, that's the single ring structure. Now, this is important too, size-wise, that a purine is always connected to a pyrimidine in order to make the two sides nice and parallel without just kind of this hourglassy figure of two little ones with two big ones. But this makes it a nice compact molecule when there's always a purine with pyrimidine. Now remember GCAP puppy 32, and that's referring then to the 3 hydrogen bonds versus the 2 hydrogen bonds. So this again is just talking about why 2 purines wouldn't fit together, why 2 pyrimidines wouldn't fit together, and why it always needs to be a purine then with a pyrimidine in my DNA. But those are my base pairing rules, and that's my basic structure of DNA. Again, remembering 5' end and 3' end. Now also remember these polynucleotides, each side, are held together by covalent bonds. But these hydrogen bonds in the middle are weak bonds. And this is really important in understanding then that we can separate the hydrogen bonds in order to be able to read the order of bases, because that is where our genetic information lies, is in the order of bases along one side or the other. Now, one side is going to be information for making a protein. The other side is just the complementary side, is what we need to make sure we're understanding. Now, in this particular chapter, though, what we're going to be addressing is we are going to be addressing DNA replication. And that's going to be the important part, because once this structure was figured out, then actually the understanding of how DNA replication came to be was actually pretty easy. Now, we needed with DNA replication, that process of copying DNA. We know that it is a semi-conservative replication. So just in general, how replication takes place is I have my 2 strands, my double helix here, but I have an enzyme that actually separates the hydrogen bonds. This now is going to serve as a template for new DNA to be built on top of it. This strand is going to serve as a template for new DNA to be built on it. So here's the side that went 5' down to 3'. Well, complementary base pairing rules say A always matches with T, C always matches with G, and so the new strand gets built with complementary bases, but again, built in an anti-parallel direction. This was my template from the 3' down to 5' end, and so I have an enzyme, DNA polymerase 3, that's going to be the main enzyme that's actually going to match complementary bases to build a new side. So this is referred to as semi-conservative because I'm always saving half old and building half new. So semi means half. Semi-conservative means I'm saving half of the old DNA and just building half new when DNA replication takes place. Now, who is credited with determining that it was semi-conservative is actually a group of scientists, Michelson and Stahl. So Michelson and Stahl, what they did is they actually used heavy isotopes of nitrogen in order to have an initial strand and kind of track with the initial strand of DNA. They knew replication was happening, but now by having an original strand of DNA that was heavier, now they could actually track when new DNA was made. They could actually predict that when I started with heavy DNA, they could actually predict that I'm going to actually have different masses of DNA based on when and what round of replication it was. Where's my heavy DNA? So this actually gave them a semi-conservative. When semi-conservative replication happens, I have my original heavy DNA. In my next round, I'm going to have half heavy, half light. And then in my next round, I'm still going to have a strand that's heavy, here a strand that's heavy, but I'm now going to have new strands that didn't have that original heavy nitrogen isotope in it. So you can actually see that in centrifuging and separating out different DNA of when it actually got made. Now, with that, just understand Michelson and Stahl, that's actually what they are credited with, is determining that it was semi-conservative. And we definitely now know that. Now, when we do talk about DNA replication, we do have to remember differences in bacteria versus eukaryotes in their DNA. Remember that in bacteria, in prokaryotes, DNA is circular. This is going to be a big deal when we start talking about telomeres, what happens in the end. In eukaryotes, remember that DNA is linear, so there's a beginning and an end to it. Now, what ends up happening in replication is there's always an origin of replication. So here's an origin of replication here in my circular DNA, And actually, in order to be able to effectively and efficiently copy our 3 billion base pairs within that S phase, there are multiple origins of replication. Now, recognize too that with that origin of replication in a eukaryote cell, it actually can replicate two different directions. And here's now another replication bubble where it can actually replicate two different directions, and another replication bubble so there's multiple points of origin of where replication begins. Now, what we're going to look at is we're actually going to look at a replication fork. We're just going to look at one region where DNA replication is taking place. And a replication fork is where an enzyme has been separating the two strands of DNA, and now where new DNA is being built on both strands. Now, what I also need to point out to you too is that when we have our free nucleotides, these free nucleotides, because this is an energy requiring process. An energy requiring process, instead of just having now ATP and all sorts of energy, well, ATP is essentially a nucleotide with 3 phosphates on it. So it's the adenosine with 3 phosphates. So all nucleotides actually have these free nucleotides already are bringing their energy source of 3 phosphates with them. And so polymerases will actually utilize these inorganic phosphates to now power this building of covalent bonds. And again, remember, these are dehydration reactions that are actually joining these nucleotides together. But it's DNA polymerases that are actually going to be building polymers of DNA. Now, in this chapter, we're going to have to learn 2 different DNA polymerases. We're going to have to learn DNA polymerase 3, but then we're also going to have to learn DNA polymerase 1 and make sure we understand the difference between them. Now, all polymerases, all primases, they can only build and add new nucleotides to the 3' end. So when DNA gets elongated, new DNA can only be built on the new strand 5' to 3'. So this is going to make a big difference when I'm looking at a replication fork and I already had my original strands of DNA, my template strands. One is 5' to 3', the other one's 3' to 5'. I'm going to have different directions of building my DNA. Because again, polymerases can only build DNA in the 5' to 3' direction, adding new nucleotides only to the 3' end of the new strand. So that means along the template strand, the template strand is anti-parallel. It's 3' to 5'. Remember, that stays fixed. But when we reference that fact of DNA polymerase can only build 5' to 3', we're referring to the new strand that's being built on top of the existing template. Now, here if I take a look at my replication fork, okay, so we're going to have to make sure that we're learning a whole bunch of different enzymes that are here. Now, first of all, I have an enzyme that actually breaks the hydrogen bonds. Now, that is helicase. So helicase is going to actually be working up along in the front to actually separate as we're replicating. What's going to end up happening is I have helicase first that breaks the hydrogen bonds. So that's really important. Now, for a DNA polymerase to know where to begin, what actually has to occur is I have to have a primer laid down. So a primer is actually RNA nucleotides that attach to the DNA. So there's an enzyme called primase that lays down the primer. Now, without having that RNA primer there, DNA polymerase 3 doesn't know where to begin. It needs that signal to go, oh, this is where I need to start building new DNA. So I have to have an RNA primer. Remember, the RNA primer is it's a short strand of RNA nucleotides that signals DNA polymerase 3, this is where to start building the new strand. So I've got helicase that unzips the DNA. I have primase that lays down a primer. And then what I have is DNA polymerase 3 that is actually going to now join at the primer. And now it's going to start adding nucleotides, complementary nucleotides, to the growing new strand of DNA. Now it only builds 5' to 3'. Now on this strand, it can actually just follow the helicase. As helicase unzips, it can actually just continuously build in the 5' to 3' direction following helicase. But on my other strand, you're actually going to see that actually DNA polymerase 3 is building just these little short segments backwards because of the 5' to 3' issue. These are actually referred to as Okazaki fragments. So these little fragments of DNA that are being built in short sections. But overall, so what's happening is helicase is unzipping. Well, it's going to build a little bit and then go, oh shoot, got to catch up. And then it's going to build until it hits that primer, and then it's going to go back and it's going to build a little bit more. But it's still trying to keep up. This is referred to as the lagging strand because it just happens a little bit slower because it has to go in short little segments in order to build 5' to 3'. And then I always have a leading strand, and the leading strand can just continuously elongate the DNA in the 5' to 3' direction. At every replication fork, I am going to have a leading strand and a lagging strand. Now, we just have to look at what is the original direction of the DNA and recognize that new DNA can only be built in the 5' to 3' direction. You're probably getting sick and tired of hearing that from me, and that's good because it needs to be drilled into your head that polymerases can only add new nucleotides to the 3' end of the growing strand, of the new strand. So new DNA is only built in the 5' to 3' direction. So keep that in mind. So polymerase 3, that's my main DNA polymerase that's going to be building my DNA. So again, in this picture, I have primase that lays down the primer. And a primer is RNA, so it doesn't last. So we're going to have to do something with that primer. We can't have segments of RNA and then DNA. That doesn't work. The RNA is not a stable molecule and would eventually fall off, and then we'd have holes in our DNA. So that's not going to work. Now, here's my primase. Here's my replication fork. Here's my helicase that's unzipping. But a couple other enzymes that are now pictured in here that we should be aware of is topoisomerase. It's a full mouthful. Topoisomerase is an enzyme that's kind of helping to unwind the DNA upstream of the helicase. So, because remember, I got this tightly wound double helix, so I've got to have topoisomerase here that is actually helping to unwind the DNA upstream. Now, what also needs to happen too is I've got to hold down my 2 strands of DNA that are now free from the helicase. So I have this other protein that's called single-strand binding protein. And single-strand binding protein is what it says. It's going to bind a single strand of DNA, kind of hold it down, so primase and DNA polymerase 3 can do their thing on it and build the new strand on top of it. So topoisomerase kind of loosens and unwinds the DNA upstream. Helicase is going to separate the 2 existing strands by breaking hydrogen bonds. And then I'm going to have single-strand binding protein that's holding those 2 template strands down while primase builds a primer, and then DNA polymerase 3 comes in and builds in the 5' to 3' direction. So what we talked about was at any replication fork, I am going to have a leading strand. So this is where my DNA polymerase 3 can actually just follow along with the helicase. But then I'm going to have a lagging strand where I have what are referred to as Okazaki fragments. So those Okazaki fragments are these fragments, and why I have Okazaki fragments is because DNA polymerase and primase can only build new polynucleotides in the 5' to 3' direction. So now I have to talk about how do I get these primers out. So this is where 2 more enzymes are going to come into play. I have DNA polymerase 1. Now, DNA polymerase 1 actually can replace the RNA primer with DNA nucleotides. So this is the role of DNA polymerase 1, is to get rid of the primer. Like, okay, we've built the majority of the DNA with DNA polymerase 3. Now we don't need the primer anymore. We can't leave it there. So DNA polymerase 1 comes in and replaces those nucleotides, those RNA nucleotides, with DNA. Now, one last thing has to happen because DNA polymerase 1 is not able to actually make that nucleotide connection to the 5' end. So I have to have another enzyme called DNA ligase. DNA ligase is the final enzyme that can now make that covalent bond here between these Okazaki fragments in order to have now a continuous polynucleotide. So in now fixing the Okazaki fragments and sealing it, well, I have to have my DNA polymerase 1. Its job is to remove the primers, replace it with RNA nucleotides, and then I still have to have ligase that now completes the covalent bond in my new strand of DNA. So here I'm seeing again my RNA primer. I've got my DNA polymerase 3. I've got my helicase. I have my single strand binding protein that's happening here. And here we're putting it all together. We're now on my lagging strand. I'm actually seeing that I've got lots of primers and I've got lots of little strands here. So this is where I need to have DNA polymerase 1 that is taking care of the primers, and I have to have ligase that is forming the covalent bonds between those DNA fragments. So again, leading strand versus lagging strand is what we need to make sure that we're understanding. So Okazaki fragments are always on the lagging strand, and why we even have them, polymerases and primases can only build 5' to 3'. So here's your overall picture. This would be a good one to just make sure that you are addressing and understanding before you take the test. You've got to know all of these different enzymes and what's going on here in DNA replication. Just double check. Here's just another diagram that's showing all the different enzymes that are involved in DNA replication. Now, polymerases don't always get it right. Mutations happen. They will put in incorrect nucleotides every once in a while, and when that happens, there is proofreading that occurs. And so proofreading DNA, and this is actually why we don't want to rush the S phase of the cell cycle, is we want to give your DNA time to now proofread and double check for errors. So a very common error that actually happens, and this can actually happen with a lot of just carcinogenic types of encounters, especially UV light, can actually create thymine-thymine dimers. And so where now the two thymines will actually form a bond together. And so that's going to cause a problem in the next round of DNA replication. So what we actually have is an enzyme that's called a nuclease. And a nuclease is actually going to come in and cut out the incorrect DNA to allow for a DNA polymerase to come in and repair the error. And then we still have to have ligase that can now seal it up so it looks perfect. So this can happen in your mismatch repair. This can happen in any repair enzyme. So these are my repair enzymes, my nuclease, and then I have a DNA polymerase that does this and ligase. Or it's an excision repair like this, where it's like, oh, nope, we have this error, cut out the bad part, replace it with the good part, is what we want to do. So this is my proofreading of DNA. Now, something to keep in mind, DNA has proofreading. When we start moving into RNA, RNA doesn't proofread. RNA will just make copies of DNA or make copies of itself, and there's no proofreading involved. So this is going to play a big role when we start talking about mutations that exist in different viruses, DNA-based viruses versus RNA-based viruses. But proofreading only exists in DNA, not in RNA. So something else I want you guys to make sure that you are understanding is eukaryotes versus prokaryotes. Because of this thing called primers, if you have circular DNA, at the very ends of your DNA, if you have circular DNA, you don't have ends. But if you are a eukaryote cell, you have ends of DNA where now there's primer that has to now start the replication process, but there's nowhere for, because you're on the edge, there's nowhere actually for my polymerase 1 to attach and replace that primer. So what actually happens in eukaryote DNA is we actually shorten. Every time our DNA replicates, it gets shorter and shorter and shorter in our cells. So what we actually have are segments of DNA at the ends of our chromosomes that are called telomeres or telomers. Who knows? Potato, potato. These telomeres at the ends of our DNA are they're just useless DNA. Like, sure, fine. We can lose those suckers. And so we can actually, it's actually a huge study within the past decade, understanding just our key to longevity is we can actually tell the age of somebody based on their chromosome because of how short the telomeres are actually getting. So when we talk about the telomeres are these ends of DNA, of eukaryote DNA only, because then prokaryotes don't have to worry about it because they're circular DNA and there's no edges that aren't going to get replicated. So telomeres get shorter and shorter with every DNA replication and every cell division that ends up happening. So here's actually just highlighting where these telomeres are at the ends of your chromosomes. But in some cells, because we can't have that happen in our germ cells that are producing our gametes, so there's actually enzymes called telomerases that actually help solve that problem. What they typically do is they provide this extra scaffolding at the ends in order to kind of elongate these telomeres and allow for replication to actually happen. So we don't end up getting the shortening that happens at the ends of each of our chromosomes. Because again, in our germ cells that are making gametes, we can't afford every generation to get shorter and shorter DNA because eventually we'd have no more. So there is the telomerase that solves that shortening of the telomere issue in certain cells. Now, last thing we need to talk about before I just do another segment on genetic engineering, just going to talk about packaging of chromosomes. So if we talk about DNA, what I do need you guys to understand, so when we talk about DNA, it's our double helix and it's just now nucleic acid. That's all it is. But in order to now get to highly condensed DNA actually has to organize itself around these proteins that are called histones. Now, I think of these histones as kind of like the spool that thread wraps around in order to keep it organized. So what will happen is these histones, and these are proteins that DNA will wrap around in order to get more organized. Now, when it actually bundles together like this, it actually bundles in these visible structures called nucleosomes. So these nucleosomes are these bundles of histone and DNA organization. But then from this, what can actually happen is they can loop together and organize and organize and organize to now become highly organized chromosomes. Now, what I need you guys to also make sure, as it's just organizing, is make sure you know the difference between heterochromatin and euchromatin. So, because when we actually talk about chromatin, there's still histones involved, but there's regions that are tightly packed and there's regions that are loosely packed in our cells. Now, the regions in our cells that are loosely packed, well, that region is actually where protein synthesis is going to be taking place. But in regions of our DNA that we don't need for that cell, it stays really tightly packed. Because why bother unpacking it if we're never going to use it? Just store it away. So recognize the degree of packing within our chromatin varies based on our cells. So I am just going to go ahead and finish really fast and talk really quickly about genetic engineering, because really we just do need to have a basic overview of genetic engineering. So when we do talk about biotechnology, genetic engineering, we are using our knowledge of DNA in order to better advance and better understand DNA, to copy DNA, to actually be able to have other organisms make human proteins, etc. So in DNA technology, one of the things that we would be doing is actually cloning DNA in things called plasmids. So this is a way that I could take a gene from a foreign cell and actually insert it into a prokaryote. Prokaryotes replicate really fast. DNA gets read the same. A couple of issues we'll talk about when we start talking about exons and introns later, but it's a little more complicated than just taking a human gene and inserting it into a bacteria. But there are these parts of bacterial DNA that are called plasmids. So these are very small circular pieces of DNA that actually bacteria use to exchange to increase genetic diversity. So plasmids are actually something that you can take out of a bacteria fairly easily, but then also make living bacteria uptake a recombinant DNA, a recombinant plasmid that now is a combination of the original bacteria plasmid and then a foreign DNA back into it. And so once that happens and you can get it activated, well, we can actually now genetically modify all sorts of organisms for benefit, like having genetically modified types of crops actually reduce pesticide use. We're actually able to make human insulin or proteins. We're actually able to genetically engineer bacteria that can clean up oil spills. And so there's all sorts of benefits to our genetic engineering and genetically modified organisms. Now, there's a lot of ethical issues and business side things that we won't get into. This is a biology class. You can do a business ethics conversation about, intellectual property and not another time. Now, the basic thing that needs to happen in cloning DNA or making recombinant DNA is you have to be able to cut open the DNA. So this is where we need to understand restriction enzymes. A restriction enzyme is an enzyme that will actually cut DNA. A restriction enzyme is actually found from bacteria. Because remember, bacteria have been under siege from viruses for their entire life on this planet. So they've actually evolved ways to combat these viral infections from bacteriophages. So they have restriction enzymes that will just chop up the DNA that the bacteriophage is trying to inject into them. Well, each restriction enzyme actually will cut DNA at a known specific restriction site. It is a set pattern or set genetic code that they will actually cut at. Now, most restriction sites, so the restriction sites on the DNA where the restriction enzyme will actually cut it, most restriction sites actually have a pattern where the DNA code on one strand is actually the exact same going backwards on the opposite strand. So here, my sequence is GAATTC, while on the other side, it's GAATTC is on the other side. When restriction enzymes cut, most often, most restriction enzymes will cut leaving a sticky end. And when we say it's a sticky end, it means that it's leaving these edges of unmatched bases. Well, these unpaired bases want to match with their complementary bases. So you use the same restriction enzyme to cut from the donor gene that you're looking to copy. So if you use the same restriction enzyme, then your sticky ends will match. And then you use ligase to actually seal it up. And with that, then we actually now can form recombinant DNA, get the bacteria to uptake it, and now bacteria just replicates it and treats that new recombinant DNA as its own. Now, some other techniques that get used are with here, what we're understanding here is gel electrophoresis, and we're actually using restriction fragments to actually separate out DNA fragments. So make sure you watch the video on just different biotechnology techniques here and what that would look like. Also PCR. PCR is actually a technique that's actually using our understanding of DNA replication to actually replicate DNA in a laboratory. And we can replicate RNA. It's actually what COVID testing right now is based on, is PCR, of just replicating the messenger RNA that is the information that is the COVID. Here we go. I gotta get shaken here before my second period comes in. So understand PCR. Know that with computers, we actually DNA sequence. This is how we match our entire genome. But then what you should also understand and do a little reading, watch what CRISPR-Cas9 technology actually is. Avoid touching your eyes, nose, and mouth. Go cats. So last thing with CRISPR-Cas9, I posted another video for you guys too, but this again is from bacteria. Bacteria actually use this as a guide to cut out DNA that was not supposed to be there. There's an enzyme that will do the cleaving, but with that, what they end up having is a guide RNA template that's complementary to the sequence that they are searching out to be able to remove. Now, just this year, two female scientists actually just won the Nobel Prize for their discovery and their use and expansion of Cas9 technology to now be very precise in actually removing unwanted genes in a genome. So pretty cool technology. So with that, make sure you guys are doing the reading. Make sure that you are following some of the videos that I asked you guys to follow. But most importantly, it's making sure we are super clear on all the enzymes that are involved in DNA replication. That's going to be the bulk of your test, is making sure you get those enzymes down on how DNA replication occurs. So with that, I know this was long, but there's a lot of terms that we needed to go through. But otherwise, I will see you guys in class to clean it up a little bit more. So talk to you guys later. Bye.